Have you heard about cisplatin? Anybody? Okay, thank you. Have you heard about cancer? Oh yeah, much better. I want to talk about the anti-cancer drug. Cisplatin is a platinum-based chemotherapy medication. In 1978, FDA approved it. Since then, it has been used for various cancers. It is very effective, still in use. But cisplatin has a significant problem called cisplatin resistance. In this case, cisplatin becomes less effective. Here's the cisplatin. You can see the PT, that's platinum atom, and then the surrounding. So if you want to improve or modify the cisplatin, you can change the metal or ligand or everything. So I changed the metal part first, no platinum, instead gold. Why? Because gold has a slightly similarity as platinum. And also gold has a long history of medicinal use. And I made gold phosphine complex like this. Au means gold. And I want to see if my new gold compound has anti-cancer effect or not. And I measured toxicity. Here's in table, you can see the many numbers. We say IC50 value. What's that? It means how much is the chemical needed killed 50% of cancer cell. In other words, smaller number means more toxic. Okay, the red box says my new gold compound, compound 1, is much more toxic compared to cisplatin. At least 13 times up to 90 times for the 3 cancer cell line. And I hired MRC5 cell in a green box. That's a normal cell because I want to see the selectivity. If my new gold compound kills both cancer cell, normal cell equally, it is just poison. As you can see, the green box says my new compound is only twice, less than 2 times toxic for the normal cells compared to cisplatin. Okay, let's wrap it up. My new gold compound is much more toxic for cancer cells, but it's just slightly toxic for normal cell. So I can say I made very potent anti-cancer effect compound with gold. Thank you.
When a story hits the popular press about a new species being discovered, I think a lot of people imagine an old school naturalist wearing a pith helmet and whacking through the jungles of Africa. But the truth is that 2 out of every 3 species that are discovered by scientists in the basement of museums who are hunched over microscopes and studying minute anatomical details. The species that I've spent the most time hunched over have been Tenrecs. Tenrecs are a group of animals that only live on Madagascar. Some of them look like shrews, some look like moles, and some look like hedgehogs. So for a long time, people assumed that they must be closely related to those animal groups. It wasn't until 1991, when scientists started using genetic data to understand the relationships among species, that they realized that Tenrecs aren't closely related to shrews or moles or hedgehogs. In fact, Tenrecs are most closely related to animals like the elephant, the manatee, and the aardvark. For the last few years, I've been studying the systematic biology of Tenrecs. Systematic biologists are concerned with generating the tree of life, with figuring out the best way to classify life on Earth, and with discovering and documenting new species when necessary. Tenrecs are a poorly studied group, so an evolutionary tree had never been made. So this was the first time that anyone had ever done it. And it's not as easy as you might think. For a tree with 32 tips, or 32 species, like the one shown on the screen, there are 9 times 10 to the 36 possible ways that those species could be related, and only one of them is the truth. So to get as close to the truth as possible, I used genetic data from 10 different genes, and I used a variety of statistical models. Once we felt like we had established a good evolutionary framework, we were able to start asking new questions, like how many species of Tenrecs are there really? My colleagues and I have been going into the field in Madagascar and into drawers in museums using scales, calipers, and digital cameras to document all of the variation in Tenrecs that have been collected. This has revealed some variation that we hadn't noticed before. And my colleagues and I have already documented 3 new species in the literature, but we think that 10 to 15 new species of Tenrecs are likely still waiting to be described. And this is important because 40% of Madagascar's forests are already gone, and that rate is only accelerating. So there's a good chance that these species could go extinct before we ever even knew that they existed. So while I originally got into this work because I wanted to be one of those old school naturalists whacking through the jungles of Africa, I've realized that studying DNA sequence data and minute anatomical structures is just as important and no less exciting type of adventure. Thanks.